In order for the cells in your bone marrow to infiltrate the blood, to add cells with long telomeres, they have to divide. Okay. And so whenever a human cell divides, the telomeres get a little shorter.
Welcome, everybody. This is Buck Joffrey with the Longevity Roadmap. Today, we have a really interesting conversation with Dr. Bill Andrews. He is a guy who actually was the guy who discovered the telomerase gene. Telomeres are a major part of the biological aging process, the shortening of telomeres. Basically, telomeres are, he'll describe them another way. The way I've always thought of them is they're sort of the ends of your shoelaces. They're the little plastic things that over time they wear out. And the shorter they get, the bigger problems you have in terms of, your cells dividing. And over time they get shorter and shorter. And then eventually, if they get too short, your cells become senescent. There was a lot of talk of telomeres early on in the longevity space before I think a lot of these other discoveries. And I think this is a really interesting conversation because I think it refocuses the importance of telomere length and the role of telomerase, which is the gene that actually can restore the length of the telomeres. This guy, Dr. Andrews, is obviously very smart. He is, he's known as, again, discovering the human telomerase gene. He's inventor of the year. All sorts of stuff. And he's convinced that, the key to the aging process, or probably the biggest key to the aging process, and one that all of these other hallmarks of aging, for the most part, kind of go downstream, is the shortening of telomeres. So we're going to get into that. And we're also going to talk about, some potential ways of reversing telomere shortening, or at least slowing it down. Anyway, for those of you who love longevity science, this is going to be a really good one. Enjoy it. And we'll have this interview right after these messages. Hey, longevity enthusiasts, it's time to take it to the next level. I've been fine-tuning my longevity regimen for years, and I look better and feel better than I did a decade ago. In fact, my blood work is even better than it was back then. And it's all because of my data-driven regimen, and it's inspired me to create a course and community just for you. It's called the Longevity Roadmap, and I urge you to check it out. If you're tired of your belly fat, tired of being tired, or just want to optimize yourself for the next 50 years, visit longevityroadmap.com. That's longevityroadmap.com. Welcome back to the show, everyone. Today, my guest on Longevity Roadmap is Dr. Bill Andrews. He's a molecular biologist and CEO of Sierra Sciences, known worldwide for discovering the human telomerase gene. He was recognized as National Inventor of the Year, holds over 50 biotech patents, and has published extensively in the field of aging and cellular biology. His pioneering work focuses on reversing age by restoring telomere length, a key factor in cellular health and longevity. Welcome to the show, Dr. Andrews.
All right. Thank you for having me on.
Okay, well, this is an interesting topic. A lot of people, you're best known for discovering the human telomerase gene. For listeners who aren't scientists, or, they just, maybe just physicians even who heard about it in medical school, but, maybe just a little bit of a reminder on a little bit in terms of what telomeres are and why they matter so much when it comes to aging.
Well, first of all, they're at the tips of our chromosomes. And I like to compare them to ride tickets at an amusement park. Every time a human cell divides, it loses a ticket. Okay. And then when the cells lose the last ticket, they can no longer divide. They go into a phase called senescence and then die shortly afterwards. And it's actually there's several clocks of aging, but it's the clock that makes the most sense in terms of we know how it functions. We know what causes it to shorten. We know to lose ride tickets is the same as telomere shortening. Telomeres are DNA sequences, and our cells lose a little bit of that sequence every time a cell divides, just like it's as if it was right tickets. But we know how it happens. We know why it happens. We know how it affects epigenetics, which is the turning on and off of genes in our chromosomes that make it so that our cells and our body look older, feel older, and behaves older. And so it's something that I learned a long time ago is something we need to focus on first, even though I do believe there's other causes of aging, which I think of all as multiple sticks of dynamite that are burning inside of our cells. But the one with the shortest fuse in humans is the length of our telomeres. And also, we know now that the length of the telomere controls all the other clocks too. So like the DNA methylation clock, the IgG glycosylation clock, which are some of the more recent tools for measuring biological age, they appear to be very likely controlled by the telomeres. And if not, nobody has figured out what controls those ones. So, okay, so they are correlated with aging, but they don't have any understood mechanism as to why they correlate with aging. Telomeres explains it better than anything.
Where does telomerase come into this, and how your discovery there?
Yeah, well, so every time our cells divide, it loses a ride ticket, or our telomeres get shorter. But if that was true in our reproductive cells, then our children would be born with shorter telomeres than we have. Their children will be born with shorter telomeres than they have. So we knew back in the early 1990s that our reproductive cells must have some way of not losing the ride tickets. Well, or when it loses a ride ticket, it's given a ride ticket back. And so in my research at Geron Corporation, I led a team that ended up discovering this enzyme, human telomerase, that is found only in our reproductive cells. No other cell of our body has it. And every time our reproductive cells divide, it does lose a little bit of telomere, but then telomerase adds it back. So I think of it like a tug of war. You've got shortening, and then you got lengthening, two sides of a tug of war. And it's a tie. So it's back and forth, back and forth. But in every other cell of our body, that telomerase isn't there. And it's because the gene that produces telomerase is shut off. And therefore, we only have a one-sided tug of war. We have just shortening. Every time a cell divides, it gets shorter and shorter.
How does that telomerase activity vary over time? I guess as we, when we're young, when we're children versus when we're, middle age and older.
What telomerase doesn't vary whatsoever. It's just completely shut off. There's absolutely zero telomerase activity in all of the cells of our body.
From day one after we're born?
When we're conceived, even before we're conceived.
Okay.
The telomerase gene appears to be shut off during the formation of the sperm and eggs that are fused and fertilized to create the first single cell embryo. So it's somewhere around the 8-cell stage of the embryo that at least one cell turns the telomerase gene back on, and then that cell becomes our primordial germ cell line, which is what produces our sperm and our eggs. So, but it's completely shut off.
Correct me if I'm wrong. I mean, I'm not an expert on this, but there's sort of a nonlinear relationship where the telomere length doesn't necessarily shorten at a steady state, right? I mean, it can lengthen or shorten in response to environment, stress, lifestyle. Is that true?
No lifestyle thing lengthens it. No therapies lengthen it. There's no diets lengthen it. The only way to lengthen telomeres is to induce the production of telomerase inside of all the cells of our body, not just our reproductive cells. And none of the things that you hear about actually do it. But, it's nobody's lying to anybody. I think a lot of people just don't understand telomere biology.
Yeah, I mean, one study comes to mind with I'm sure you're aware of this. There was the one with Israeli studies in the hyperbaric or
Yeah.
uh,
There was just a
oxygen.
big article about that in Scientific American. Um, okay, so here's the thing, there's YouTube videos that people can watch, and I can send you those YouTube videos where I explain what's going on here. Okay, so what they're measuring is before and after. They're measuring telomere lengths before and after, and they're finding that telomeres are longer after.
After hyperbaric oxygen, just so people understand what we're talking about here.
I actually don't want to promote any products or anything like that.
Sure.
You can say it.
Sure. There's basically this Israeli study that I'm referring to, and I don't have it in front of me, but broadly, it's pretty. I mean, it's often referenced in this longevity space. Basically, the idea is that over this period of time, this very rigid and lengthy hyperbaric oxygen schedule resulted in telomere lengthening. So that's what we're talking about. But sorry to interrupt. Just wanted to give context.
Yeah, but it's not actually telomere lengthening. It's actually the telomeres are longer, but they didn't get lengthened. And that's, I understand telomere biology extremely well. I'm an inventor or co-inventor of every assay for measuring telomere length and measuring telomerase activity. But what's happening here is that if you do anything to your body that is toxic, let's say, that can preferentially kill off the cells that have short telomeres. And inside of our blood, we have cells with telomeres of all different lengths. Okay, some cells have long telomeres, some have short telomeres. So anything that's toxic is going to kill off the cells that have short telomeres. And as a result, your average telomere length is going to be longer. Okay, just as if you had a company and you fired your dumbest employees, the average IQ of the company goes up without anybody getting any smarter. And very few people understand that principle. Okay. But the other thing is that if the product or therapy or whatever is immunogenic at all, inducing an immune response, that's going to drive cells that are in the bone marrow that have already long telomeres. Even if you're 90 years old, you have cells in your bone marrow that have long telomeres. It's going to cause those cells to start dividing and infiltrate the blood. And that, again, is going to cause the average length of your telomeres to be longer without any lengthening. Just as if your company hired a bunch of geniuses, the average IQ of your company increases without anybody getting any smarter. Now, the only way to actually show that you actually had lengthening is to show induction, that you are actually producing telomerase in the cells. Because as I said before, that's the only way to lengthen telomeres. And I'm not aware of any of these companies ever showing that kind of data. And I'm suspicious. And by the way, we test everything here, and we haven't been able to demonstrate telomerase activity with any of the methods that are available. But we think they probably did try to measure, because that's an obvious thing to do. And the assays are easy. If you're lengthening telomeres, show that you're producing telomerase. And if you haven't, then why not? Okay. And that's what people should ask when somebody tells them they have a product that can lengthen their telomeres. They should ask, does your product induce telomerase? And if they say no, then their product does not lengthen telomeres. And the next question is, why not? Since it's easy. And but I would not believe it. Not that they're lying. I'm just saying that they misunderstand it themselves. Their product isn't lengthening telomeres. But the big problem is that when you do kill off the cells with short telomeres or add new cells with long telomeres, that requires additional cell division in the body. And so even though the telomeres in your blood are longer, the average telomere length of your whole body is shorter. Okay, so it's actually accelerating aging. So a lot of these products are accelerating aging. The only product.
Explain that, if you would, just explain that part again, because in my mind, I mean, I'm following, like, all of a sudden, I totally understand your metaphor there. You're essentially knocking out, these shorter telomere cells, and so the average of cells there becomes longer telomeres. You're recruiting from bone marrow, you have cells there with longer telomeres. So explain to me how that accelerates aging.
In order for the cells in your bone marrow to infiltrate the blood, to add cells with long telomeres, they have to divide. Okay. And so whenever a human cell divides, the telomeres get a little shorter. So even though they're already longer than the telomere length in your blood, when they divide and produce a lot of cells that then infiltrate the blood, those cells have longer telomeres. But now the cells in your bone marrow are shorter as a result of that cell division. So the average of your whole body is actually shorter. And anything you ever do that induces cell division in your body is going to cause telomere shortening.
Got it. Got it. Let's talk a little bit about the I guess the potential ways that you can attack this problem from from the standpoint of what you're doing in your lab. If we know that, telomerase is just not there once conception, basically, what can we do to attack this problem?
You can do things to slow down the rate of accelerated telomere shortening. Okay, so before, when about 10 minutes ago, you mentioned something that lifestyle and stuff like that can affect telomere length. It can't lengthen it, but it can sure accelerate the shortening. And that's like anything that's like smoking, obesity, psychological stress, oxidative stress, inflammation, all those things will accelerate telomere shortening. And if we do the math on the rate of telomere shortening without doing anything, with having the perfect lifestyle and perfect genetics, our lifespans are limited to 125 years. It's the only hard brick wall that anybody can run into in terms of extending their lifespan. You cannot exceed 125 years because of telomere shortening. But nobody ever lives to be 125 because we all accelerate our telomere shortening one way or another. But in our research, what we are doing is we are trying to find anything that will cause our cells to produce telomerase. Now, and let me say in a really short term, and there's YouTube videos if people want to hear more about this, but the reason why we shut off the telomerase gene in every cell of our body is because there's a benefit to our species, but not the individuals. There's a benefit to our species to eliminate the longer lived people. Anybody who's lived long enough to raise their young, they do not contribute to the success of the species anymore, because their best way of contributing to the success of the species is to shuffle their genes through sexual reproduction to create offspring that have new diversity, variations that didn't exist before. And the more diversity that exists in a species, the more likely that species will survive rapidly changing environments, or at least some members of the species will survive rapidly changing environments. So any species that evolved an aging process, any species that had sexual reproduction and then evolved an aging process was more likely to survive rapidly changing environments than a species that didn't. If people understood what I just said, that's great. But if they didn't, there are YouTube videos where I spent an hour at least on explaining that specifically.
Sure. When we talk about, the one thing I think about is, okay, well, I presume this issue with telomerase is across species, right? So even in whales and
no, actually not.
Okay.
It's aging is a relatively recent evolutionary event, and so many species have evolved different ways of eliminating the longer lived. Okay, humans, dogs, cats, horses, sheep, pig, and deer are the only animals that have ever been shown to eliminate the longer lived by telomere shortening. Rodents, mice, rabbits, things like that, they do not age by telomere shortening. They use other mechanisms, most likely oxidative stress and mitochondria dysfunction, to eliminate the longer lived. But some animals, like you'd mentioned, whales, humpback whales, lobsters, tortoises, clams, they don't have any aging process, any detectable aging process at all. And at least nobody has found any yet. People didn't start looking until the time of Darwin, and the only way to know is to have them be there when they're born, and then watch them as they age. And almost none of them have the equivalent of rings on a tree that you can count to figure out how old they are. So you don't know how old an organism is unless you're there when it's born and you watched it its entire life. And then 200 years later, there's still animals that are showing no signs of aging. Now, the exception is clams. Clams do have stripes on their shells. They get a new stripe every year. And so a lot of people have started counting the number of stripes on the clams, and there are clams over 500 years old. Okay. People didn't know that lived that long. And these clams, they have, and all these animals I just mentioned, lobsters were the first one that I identified, but they all have telomeres produced in all the cells of their body, unlike humans. They don't have any telomere shortening at all. And they never get cancer or any kind of diseases, are extremely rare. And they have no aging, no detectable aging whatsoever.
So that you're talking about not just clams here, but for example, that whale example I gave. So you do have telomerase. Yeah. You do have telomerase activity after while the organism's living in those humpback whales.
Yes. And they have telomerase produced in every cell of their body.
And it's interesting because, so I guess that idea then, if you're thinking about approaching this for humans, is, as you said, trying to figure out if there's anything that can trigger telomerase activity. Is there anything promising right now?
Yes, there is. Actually, we've been doing this for 20 years and we've had a lot of success. We haven't come up with anything potent enough to actually produce enough telomerase to reverse aging. But we have things that will stimulate the production of a significant amount of telomerase, just not enough to like win that tug of war that I was talking about, or even tie that tug of war. Now, see, the problem is that, as I was mentioning before, there was an evolutionary advantage to eliminating the longer lived after we raised their young. We do that there's a protein that binds to what I call the dimmer switch. Every gene has a dimmer switch, and they're called promoters that turn genes on and off. And this protein binds to the dimmer switch for the telomerase gene and shuts it off. So we are looking for nutraceuticals, plant extracts, fractions of plant extracts. We've tested over 20,000 different, or close to 20,000 different plant extracts now. And we're looking for anything that will actually dislodge that repressor protein that's shutting that dimmer switch off to actually turn that dimmer switch back on. And so we have like high throughput robots here, but that do an assay that's called the high throughput robotic mRNA RT-PCR assay. And it's a way of detecting production of telomerase in cells in a high throughput way. So these robots they don't look human. They're actually in the room directly behind me right now. They take up a whole room, but these things can test up to like 4,000 different plant extracts a day. The hard part is actually preparing that many plant extracts a day. So we have found close to 60 different fractions of plant extracts that do induce telomerase. And
in humans?
In humans, yes.
Okay.
We've actually only tested it in humans so far, but I'm hoping it works on dogs, cats, and horses too. We licensed the top 5 to a company called Touchstone Essentials that created a product called Telovital. I'm not into marketing, but I want to explain that the science there is that there is in that product, there are our top 5 telomerase inducers mixed together. And so by taking that product, you are inducing the production of telomerase, but not enough to win the tug of war. But it's still anything that slows down the rate of telomere shortening, slows down the rate of aging. And so even though I said that we have this theoretical maximum of 125 years, if we have perfect lifestyle, lead a perfect lifestyle and have perfect genetics, this product has the chance, and it's the only product that has the chance of extending this beyond that 125 and delaying, not just extending lifespan and health span. because we know from studies of gene expression and stuff and the role of things called enhancer sequences and stuff like that, the telomeres actually play a role in expressing all the genes that are involved in aging, wrinkled skin, hair color, things like that. All these things can be slowed down by doing anything that induces telomerase enough to lengthen telomeres a bit, but not enough, as I said, to win the tug of war. Let me, before you go to anything else, let me qualify that we do know, and this is well published by other scientists too, we do know that when telomeres get critically short, they become a lot easier to lengthen. So the tug of war can be won when the telomeres, by the lengtheners, when the telomeres are really short. So we suspect that people that do have critically short telomeres in either their whole body or some tissues of their body, they are going to see some age reversal because of the lengthening of the critically short telomeres. And I'm going to be honest with you, in meeting with a lot of people that have been taking products that lengthen telomeres, the people that are showing the best results are the ones that are in their 90s and stuff like that, which I'm just blown away with how much change I see them week after week after week after week. So I believe that we are lengthening their shortest telomeres.
What kind of, I'm sure you're probably referencing some small clinical studies. Can you talk about those?
Since these are plant extracts, and all the ingredients are things that people already eat anyway, that they're fractions. So we had to eliminate some of the molecules in the plants because there's also things that will inhibit telomerase activity. So if you were to take the whole plant, you might find that maybe you are inducing the expression of telomerase, but something else in that same plant is inhibiting the activity of telomerase. So we've had to do fractions, like fractionate, separate molecules in a plant by size or charge or something like that to separate the inhibitors from the activators. And so those are in there now.
It seems like this is kind of because of the nature of telomeres or telomerase specifically in different species. It seems like a very challenging area to do longevity research. I mean, I'm just thinking about the interventions testing program, for example, as I'm sure you're aware. I can't remember if you said that rats have this issue with telomerase or not, but
rats have telomerase produced in all their cells. They have no telomere shortening.
Yeah. So, I mean, in that regard, you're never going to see any potential benefits in animals. And can you talk a little bit about those challenges in this space?
Well, first of all, yeah, it's really challenging, but my mission isn't to create a product that's going to make me a lot of money. My mission is to cure aging, my own aging, especially. I
get that, but I guess I'm just trying to figure out how do you measure it?
So I would never use mice or rats or even there's another animal that's often used, a rodent that's used for studies. Rabbits either, but that's not the one I'm thinking of. Naked mole rats is the other one I was thinking of. But if we were going to do animal studies, we would use pygmy marmosets, which are primates, non-human primates, very close to humans, and at least they've been shown to age the same way humans do, and they're the size of mice, so it would make it easier to do clinical studies. But are preclinical studies in this case, which are animal studies. My company, I run the company. I don't want to spend a lot of time doing animal studies on intermediate products. Our goal is to find something that's going to actually reverse aging. And when we find something that can reverse aging, at least in human cells in a petri dish, then I want to spend all, because it's a lot of money and a lot of time to do the animal studies. And we're discovering things new all the same so frequently that before any study gets done, we're already going to have a better ingredient. So I'm waiting for something when we find something that's potent enough to win that tug of war. And then we want to do preclinical studies with pygmy marmosets, and then treat humans. And if it turns out that the product that we discover is a plant extract, because of the FDA's ruling on plant extracts being generally regarded as safe, we don't need to do the animal studies, but because of the fact that I'm more interested in curing my own aging than anything else, I do want to do these animal studies and get as much knowledge about what's going on as possible.
In the big picture of how we think of, or I guess the paradigm that a lot of scientists use, the hallmarks of aging, like mitochondrial dysfunction, cellular senescence, obviously senescence can be downstream, epigenetic drift. Do you think that telomere shortening is upstream of all of these?
Yes, absolutely. I think telomere shortening controls all the hallmarks of aging. Um, because even with all these hallmarks of aging, nobody's explained the mechanism that these things happen, except for telomeres. Telomere shortening is the only thing that has a well-defined mechanism to explain everything about aging. Um, I'm trying to think if there's any particular hallmark of aging that I wanted to address specifically, but it's
Like
pretty much
maybe epigenetic drift is
well.
curious.
Epigenetics is completely controlled by telomeres. I mean, there's other things that control epigenetics too, but the genes that are specifically involved in the aging process, those are very likely controlled by the length of a telomere. Okay, so let me quickly define what an enhancer sequence is. Enhancers are well-known things in our DNA molecules that they can be like 10,000, 100,000 bases away from a dimmer switch or promoter. And the DNA can fold over and bring these enhancers in contact with that dimmer switch and turn it on and off. Telomeres have all the hallmarks of enhancer sequences. And so they can fold over and affect the dimmer switch of genes on your chromosomes. But the difference between telomeres and regular enhancers is that telomeres shorten. And so when they shorten, they can't reach as far. And therefore, we see changes in the gene expression with age, well, with telomere shortening. So the shorter the telomere gets, the more changes you see that are consistent with becoming older or longer lived. And lots of studies have been done now where we can show that relengthening the telomeres actually reverses these epigenetics in every way imaginable. We've shown this in human cells grown in petri dishes. We've shown this in human skin grown on the back of mice. And we have shown this, and a lot of this is done in other labs because my company does not work with mice at all. But we've provided the technology to other labs to do this. Next one is people have engineered mice to make them so they do age by telomere shortening. And these mice are incredible because they have all the same human-related aging characteristics. And when the telomeres are lengthened in these mice, age reversal occurs in every way imaginable, including, this is a big surprise, memory coming back. So mice that had forgotten how to go through a maze and have symptoms similar to Alzheimer's or other forms of dementia, when the telomeres got lengthened, their memories came back. And now when I speak at conferences, at medical conferences all over the world, and on all subjects, not just aging, but when I'm at a conference on something like Alzheimer's, I hear people now saying that Alzheimer's isn't necessarily the loss of memory, it's loss of access to the memory. And lengthening telomeres is restoring that access. So the memory is still there. I actually have a clinical study in clinicaltrials.gov ready to start testing to see if we can bring the memories back in Alzheimer's patients and actually save their lives. The unfortunate thing is that we are using the only thing that really does lengthen telomeres, and that's a technique we've been using for 30 years to study telomeres and telomeres. That's gene therapy. So gene therapy is something that we want to do, but it's not the kind of thing anybody wants to be doing yet because it's still in clinical studies, super, super expensive. But at least it can provide us with proof of concept by seeing if we can save some of these people that have Alzheimer's.
So, I know we were talking earlier about nutraceuticals, but can you talk a little bit about the idea of gene therapy and like what in that situation? Would you be using various CRISPR technologies or whatever and delivering telomerase, or how would that work?
Well, CRISPR only edits genes, okay, and There's also other methods for editing genes, such as zinc fingers and talons that existed even before CRISPR. But what we want to do is we want to deliver, and we do CRISPR and talons and zinc fingers, but what we think is the most likely to work is delivering a new gene to the cells that has that dimmer switch permanently on the full strength on position. Okay, so we're delivering a new gene, but gene therapy is risky. It's doing clinical studies. It's great for animal studies. In our clinical studies, we are only going to be treating people that have like less than a year to live because of like Alzheimer's or something like that. And we're hoping that we save them. And the side effect will be seeing that they get younger in every way imaginable.
Sure. In terms of just thinking about some of the dangers of gene therapy, I guess one of the, I don't know, there's critics who talk about, when you upregulate, the potential of upregulating telomerase activity, or, just creating telomerase activity, I guess, is the potential link to cancer. What do you say to that?
Well, I was National Inventor of the Year in 1997 for my cancer research. I do understand cancer a lot. And I've invented like at least 5 different treatments, therapies, drugs that are been through clinical studies and are now available for use. And some of those are actually dealing with inhibiting telomerase. But here's the thing is that there's a rumor going around, and it's only hearsay and rumor. There's absolutely no data whatsoever saying that telomerase can increase the risk of cancer. But there's an incredible amount of data showing that it decreases the risk of cancer. And it's actually short telomeres that cause mutation rates to skyrocket that cause cancer, because all cancers come from mutations. And then it's the short telomeres that actually allow cancers to sometimes survive the treatment and then come back. Okay, so if you're being treated with a drug to kill the cancer, you can see the cancer disappear. But if the telomeres are short, the mutation rates are going to skyrocket still, and the cancer is going to mutate to a form that can come back and be resistant to the treatment. And so one of the examples is a drug called Rytelo, Rytelo, that just got approved by in clinical studies just a few months ago. I'm the inventor of that, and I argued all the time that that's going to give short-term survival, but the cancers are always going to come back. And that's why it took so long to get that drug approved, because that turned out to be right. So, but I'm very interested in coming up with ways of treating cancer by poisoning cells that are producing telomerase. Well, you're not taking a telomerase inducer, that is. But the bottom line is telomerase decreases the risk of cancer, not increases it. It actually boosts the immune system, and the immune system is our best defense against cancer. Boost the immune system by lengthening telomeres in our immune cells. And the way that telomerase gets produced is by cancer. So it still has to become cancer first. And then because of all the cell division, the telomeres get critically short, and then the mutations induce the production of telomeres. Okay, so cancers they want to live longer just like we do, okay? But if you can produce telomerase before the cell becomes cancer, it actually decreases the chance that it'll ever become cancer. And so there's actually another video where, because a lot of people used to be saying that telomerase causes cancer, but you don't find anybody saying that anymore. In 2017, I gave a presentation in Tokyo. I mean, it was a really big presentation. Only doctors were invited to be in the audience, but there was like 500 doctors. When I got off the plane, I saw my picture on billboards and stuff like that. It was a really big thing, but there was on the news somebody claiming that telomerase causes cancer. So I actually added something like 45 minutes to my presentation just to review all the data and all the facts about why some people believe telomerase causes cancer when it doesn't, and then all the studies that have shown that it doesn't cause cancer. I went through all that. And now I never, ever hear that. That was such a successful presentation, and it's available. That was such a successful presentation. I've never heard anybody say that telomerase causes cancer again, unless there's somebody who's so far removed from the field that they just remember the old rumors that existed 8-9 years ago.
How optimistic are you about, with the things that you're doing that we can, with gene therapies and advancements and AI and all this, that we can come to some kind of a solution or find some sort of method, to solve this problem of aging?
Well, let me say first, no matter what else we do to cure aging, aging will never be cured unless we also solve the telomere shortening problem. So that's why I'm focused on that, even though I don't believe it's the only cause of aging. But so I'm about 80% certain. Let's say you were asking how optimistic I'm 80% certain that when we have something that can lengthen telomeres, we will show a reversal of aging and life extension and health span extension in humans. Now, when that happens, we might not find it's perfect. We might find that there's other things going on, but that's why I'm also working on mitochondrial dysfunction, oxidative stress, things like that, because if telomeres, lengthening telomeres don't work to cure aging all by themselves, and we have to combine it with something else, I want to be certain that we're ready for that. And I would say mitochondrial dysfunction is the most likely thing. And it turns out that, you can do things like there's mitochondria turnover when the cells can dispose of dysfunctional mitochondria by what's called mitophagy. Some people pronounce it mitophagy, but I've been saying autophagy instead of autophagy for 40 years. So I'm not switching, but the words are different. But in order for the cells to produce more mitochondria, telomeres have to be long, because 90% of the genes, that 90% of the proteins of a mitochondria are encoded by the nuclear chromosome. And we have shown, or others have shown, that key proteins such as PGC1 alpha, 1 and 2, they are regulated by telomere length. So the telomere length reaches over, turns that dimmer switch on and off our nuclear chromosomes. And so when telomeres get really short, we can't produce PGC1 alpha anymore. And so we can't make new mitochondria when telomeres are short. So whatever we do to dispose of dysfunctional mitochondria, that's not going to be a benefit unless we can also produce new mitochondria, called mitochondrial biogenesis.
When those mitochondria, the new mitochondria come in, are those typically with normal, not shortened mitochondrial DNA, or?
But mitochondrial DNA is circular, so it doesn't have ends. So, but they're just as healthy as any mitochondria ever, unless you have mutations. Even if you had a mutation to the mitochondria, that would be a reason to actually dispose of that mitochondria. But so it most likely is the answer.
Got it. Tell us about where people can find those videos that you were talking about.
I'm just a medical researcher. My company is 10,500 square feet of lab space. I'm in the only office right now that exists here. So we don't really work on keeping our website up to date, but I can send you a list of like all the key videos that include what aging is, why we age, how not to age, and like all the subject about cancer. And we're also, since we're not a academic lab, we're not controlled by the publish or perish rule. So a lot of the data we have is not published, but we presented it, it's all public. We present it publicly and stuff like that, and mostly by the videos. So the videos are the best place, in my opinion, to really learn what, why, how, and how not of aging there is.
Got it. there's a product called Telo Vital. Is that, can you tell us a little bit about that?
Well, that's the product that Touchstone Essentials is now marketing, that contains our top 5 telomerase inducers. When we add each of these to cells, human cells, it causes those cells to produce telomerase. And so, in my opinion, there is nothing on the planet that's better for somebody to be doing to slow down their aging. And there's other things they could be doing in addition, such as antioxidants, anti-inflammatories to decrease accelerated telomere shortening. And then there's things that they can do, like taking nicotinamide riboside or Mitopure, urolithin A, things that really benefit their mitochondria and energy production. Because until we get something potent enough to actually win the tug of war, you're still going to benefit from the other things that cause aging. And mitochondrial dysfunction is the key, and oxidative stress, which is very associated with mitochondrial dysfunction. And then anti-inflammatories. Anti-inflammatories are really important. But there's also lifestyle, whatever. If we want to switch to that, we can. Okay, so there's a lot of things that people can do to slow down their aging. They just can't slow it enough to overcome this theoretical maximum of 125 years. And unless you take something like Telo Vital. But those include, like, let's say one of my favorites is exercise, endurance exercise, whether it's running, kayaking, bicycling, whatever, skiing. Endurance exercise is kind of like a weird thing because it's got like a Goldilocks effect. There's such a thing as too much and too little. And in the middle, there's a way of actually decreasing inflammation a lot. Okay, now when I say too much, I don't mean how many hours a day you exercise. I'm talking about how hard you exercise, how hard you push it. So I always say consistent endurance exercise is very important, but you should do it every day. But quit when it quits being fun. When it quits being fun, stop, save it for another day, because if you keep pushing it, that's going to cause inflammation and it's going to cause accelerated aging. So, the examples I use, there are a lot of really great runners that, win marathons and things like that typically run every 2 weeks. They don't train at all hardly. They just have good genetics. And so they don't need to train as hard as some people, but they run every 2 weeks, and then they do well in the marathons. They'll oftentimes cross the finish line on their hands and knees throwing up. But even worse, they'll be stiff as a board for 2 weeks because of all the inflammation. The body thinks, oh my God, it's being attacked by some virus or something like that. And so it sends inflammatory cells throughout the bodies that accumulate, especially in your joints. And so you're stiff. But if you're somebody who does endurance exercise every day, always keeps it fun, even during races, if the race quits being fun, quit or slow down. If you don't, because you're going to start inducing a lot of inflammation and accelerate your aging. So that's number one. Exercise, I think, is the key thing. And like I used to hold the world record for the most 100-mile races run in a year. I'm an ultra marathon runner, and I was always checking my C-reactive protein and other inflammatory markers before, during, and after some races. And because of the attitude that I took of just keeping it fun, I never had inflammatory reactions. And I could run 100 miles and the next day go out and run 50 miles because I wasn't stiff. Okay, so the other things that you can do are yoga and meditation. I've been the keynote speaker at several yoga meditation conferences, showing data, showing the people that meditate, because yoga is really just a form of meditation. People that meditate actually have longer telomeres than people that don't, because they decrease the psychological stress, which causes oxidative stress and inflammation. So yoga and meditation is really important. Losing weight, don't smoke, drinking, drinking especially kills liver cells. And that's okay when you're young because you have other liver cells that can divide to replace the killed cells. But now, since you're killing liver cells at a faster rate than normal, your other cells are dividing faster than normal, and therefore telomere shortening is occurring faster than normal. And so you run out of cells to divide to replace the dead cells by the time you're in your 40s or 50s, and you start experiencing liver cirrhosis, which can kill you. Liver cirrhosis is really just the accumulation of dead cells because nothing's kicking those dead cells out of the way anymore, because the telomeres are too short to do it. Same with genetic variations, such as muscular dystrophy. The dystrophin gene actually causes muscle cells to die sooner, and therefore other muscle cells divide to replace them. And that's why people don't experience the symptoms of muscular dystrophy when they're young. So when they get their 40s, 50s, they suddenly start experiencing the symptoms of muscular dystrophy. And there's these, along with a lot of other things, are ways of eliminating the longer lived. So it's beneficial to our species to not evolve a way to not age, not have muscular dystrophy, not to have other things that cancers, heart disease. So because typically all those diseases occur in the longer lived after you've raised your young. And so it's a benefit to the species survival in rapidly changing environments to eliminate you, which we're smarter than evolution now. So I have no intention of letting evolution control my life and health span.
Just out of curiosity, when you started taking that Telo Vital, did you notice anything? I mean, I know it's at the cellular level, but just pure curiosity if you've noticed any changes.
Yeah, no, I'm not a big fan of anecdotal data, but there is a lot of anecdotal. I wish we had studies going on where we were measuring a lot of different things, but because these are plant extracts, and because I'm more interested in spending my time finding even better, more potent plant extracts and trying to characterize the ones that exist now. We aren't doing those studies, but the FDA has already said they're generally regarded as safe, so they can be marketed. But I have seen my vision get better. I have seen my endurance get better. Like every week I meet with people that are taking the products to, answer questions for them and stuff. And I am getting a lot of feedback, especially from like a 94-year-old and a 101-year-old that's on the products. I just can't believe they don't have to say anything. I just see the differences when I'm talking to them on these Zoom calls that I do. And that's because they must have critically short telomeres throughout their body, and their critically short telomeres are getting lengthened, even though the product's doesn't produce enough telomeres to lengthen all the telomeres. It is lengthened the shortest ones. Now I have heard people and seen it. I've seen some people, their hair is coming back, their hair color is coming back. That can only be true if the reason they lost their hair or their hair color changed because of telomere shortening. And so there's a lot of other reasons why that could happen. So people aren't going to see it if they lost their hair or their hair color lost their hair color because of something besides telomeres. Trying to think of other things. Endurance, I think I mentioned that. I've seen my endurance get better. But again, it's anecdotal, and I would love to do a study but it's really hard to do. And the study would probably be better with pygmy marmosets because of the fact that it's hard to get humans to be completely controlled. You need to have identical twins where the twins were raised separately in different environments and they were very consistent in order to really do the study properly. That just doesn't exist.
Well, thank you so much for being on the show today. And again, this product is Telo Vital, and we'll put a link in the show notes to that. But I do appreciate all your time and all this really great information for us.
Okay, and I'll send you the videos in a few minutes.
Fantastic. Thank you so much.
Thank you.
Thanks for listening. A quick reminder that while I am, in fact, a surgeon, nothing I say should be construed as medical advice. Now, make sure to include your physician in any medical decisions you make. And also, if you're enjoying the show, please make sure to show your support with a like, share, or subscribe.